So good morning, everyone. I am extremely excited today to have a very special guest with us all the way from the US and from NASA in the US, our very favorite space agency. So this is Dr. Mark Ott, and I'm just going to read out a little short bio and then hand the floor to Dr. Ott to talk to us. NASA Johnson Space Center Microbiology is responsible for mitigating infectious disease risks during human spaceflight and providing microbiological support to maintain sustainability of spacecraft and operational systems. As the lead of the NASA JSC Microbiology Laboratory and chair of the JSC Biosafety Review Board, Dr. Ott oversees efforts to mitigate microbial risks that could compromise astronaut health and spacecraft integrity during human spaceflight, development of spaceflight requirements, and microbiological monitoring of spaceflight foods and spacecraft environments. He has published extensively in the areas of microbiological ecology in spacecraft, human and microbial responses to spaceflight, and the development of advanced tissue culture models to investigate infectious disease. So thank you again for coming along today to talk to us, and the floor is yours. Thank you.
Thank you very much for that kind introduction. I wanted to start out, and I appreciate it that there may be a lot of people in the audience who are thinking microbiology. That's not the first thing I think of when I think of NASA or spaceflight. And when you see a picture like the one I'm showing right now, that doesn't help. It's a beautiful picture, but microbiology really just doesn't come to mind when you see that. And even as NASA plans to go from low Earth orbit back to the moon, you're seeing people in a few photos like this. But again, microbiology doesn't really ring true. And in my world, the picture I like to show to describe it is this one. These are the astronauts inside the International Space Station eating a meal, happens to be pizza night. But you can kind of tell when you're looking at it, we've got food, we've got crew, we've got an environment that has a lot of nooks and crannies. It has a lot of the wiring on the outside of the wall, as opposed to what we find in our own homes. So all of a sudden, microbiology makes a lot more sense. So as I was mentioning, we're seeing a lot of microbiology here, and so all of a sudden it becomes more important. It's not only important to NASA, but as we start to look at commercial groups, and you see some of the names you may have recognized, like SpaceX or Boeing, that you've recognized that they are going to be flying up their own private missions to different spaceflight locations. They need to be able to handle any of the kinds of microbiological problems that NASA's been working with for years. But as I was talking earlier, it's going to really expand a lot over the next few years. So Micro has a lot of quote unquote customers that we're dealing with. And if you look at all of the different areas, it gives us a lot to do. We've got things like astronaut health. A lot of people instantly think of that. Or there are pathogens in spaceflight foods. We need to make sure it's safe. But things like life support system, the spacecraft integrity, containment of human waste or human trash, hygiene activities need to be controlled. And one I'm not going to talk a lot about today, but we're starting to hear more about as we talk about going to Mars, is planetary protection. We don't want to contaminate another planet as we look for life on Mars or other areas. So there are a lot of different aspects to microbiology. But I'll start off with one most people think about is astronaut health. And the big question I always get is, do astronauts get sick? Do they get infectious disease? The answer is yes. Sometimes it's hard to tell in flight because a lot of the diagnosis is based on symptomology. The crew member tells the doc he's talking to, I've got a headache, or I've got a rash, or I've got diarrhea. And you can imagine how hard it is to make a diagnosis based on, I've got a headache. But we do identify certain things as having happened, and we believe that they're accurate during spaceflight, including astronauts have had respiratory infections, urinary tract infections, ear infections, herpes zoster, a lot of rashes and skin disorders, and even gastroenteritis. To give you an idea of how much, there was a great survey done on space shuttle astronauts, and they found that about 1.4% of all medical events, not including a lot of the rashes and skin and subcutaneous infections, They're a little harder to diagnose. 1.4% were infectious disease. Now, what you may read in the literature is we have evidence of immune system dysfunction. We also have evidence of alteration in microbial virulence, which I'll talk about later. But my big question has always been, why do exceptionally healthy astronauts in a pristine environment where we are doing everything we can to stop infectious disease, why do these guys still contract infectious disease during a mission? Now, switching gears a little bit, another topic we get involved with is spaceflight food. And if you go back to the 1960s and the Mercury program, spaceflight food didn't look real pleasant. It kept people alive, but it wasn't really pretty. As we got into more modern programs like the one on the International Space Station, you can see a lot of the food, a lot more diversity, a lot tastier, and we've really developed it. And as we go into exploration mode, to the moon, Mars, we're looking at new things like growing crop. Now, to give you an idea of kind of what spaceflight food encompasses, it really is a huge list. We have fresh foods that we send up with missions. We have irradiated foods, thermostabilized foods, and one we categorize as non-thermostabilized, which means it hasn't been heat treated. Now, the one most people think about are dehydrated or freeze-dried foods that we add water to. That's the big one. But it also includes things like tea, food bars, cheese. And with everything that goes up in flight, we go to the lot, each particular production lot, and we test each one of those. And we have strict requirements. If they don't make it, we kick that lot out. Because we found a lot of microbes that could cause problems, like Salmonella typhimurium, Staph aureus, Klebsiella, and fungi, toxigenic fungi like Aspergillus fumigatus or flavus. We also have a category of food where we just take it, empty it out of one package, and put it into a spaceflight package, like the chocolate covered or the chocolate candies that the astronauts eating right now. We just repackage those and send them up. So as we start to look ahead in spaceflight food, what kind of challenges are we looking at? And you really want to produce something that's nutrient rich, but also tasty so the crew would want to eat it. Because if they don't want to eat it, they're not going to. And it has to be safe and stable for long duration missions like to Mars. So we're always looking at new concepts to diversify the food supply. And that's when we do look at things like pick and eat crop, such as lettuce or peppers that are grown in flight. Perhaps that's how we'll be doing things on the moon and Mars. We also start looking at new types of food. We get a lot of requests from the astronauts themselves saying, hey, I've got this probiotic shake I drink. Can it fly? So we do a lot of testing on those. And the commercial groups are coming up with some amazing requests on products that we've never even thought about it. The concept of eating caviar in spaceflight is probably going to happen through the commercial groups and not NASA. So the big question is, how do we continue to keep space travelers safe as all these new things are being implemented? And if you take a look overall, we've got the crew covered. We're talking a little bit about the crew and their food. But what about the spacecraft itself? And I will let you know if you're going to build your own rocket, do everything you can on the ground to prevent anything from going up. It's much cheaper and easier to handle. But when you're designing these new spacecraft, you want to have like habitat surfaces that aren't conducive to microbial growth. We've also found that these high efficiency particulate air filters that really screen the air very well are great at keeping the microbial counts down. And even with something like potable water, and you may be thinking, yeah, don't you guys recycle this water from humidity and urine and make it water again? And we do, but it's a really stringent process. In fact, on ISS, when we recycle water, it goes through a catalytic oxidizer, basically heated up to 267 degrees Fahrenheit for 10 minutes. It's disinfected with iodine. The iodine's removed, and then it's passed through a 0.2 micron filter. So this is really clean water. And just in case you were wondering, on the left, that's actually one of the HEPA filters on ISS. So they aren't small units at all. They're very large, and they clean the air very well. And looking at operations, we do a number of things to prevent infectious disease in flight. Perhaps the best one was due to lessons we learned in the 1960s. And we have what's called the Health Stabilization Program. And most people think of it not only as a quarantine, like about 14 days of quarantine to keep people from spreading the flu or the measles or mumps, but it's also an awareness program to tell people if you're not feeling good, don't interact with the crew. And it's been a really successful program. Pre-flight, we also do disinfection and microbial monitoring. We also, when we get people up there for long periods of time, we have a cleaning and cleanup remediation program. That includes things like regular housekeeping, disinfection wipes, and if the water system has a problem, we have iodine to add to the water. But even with all of that work, contamination will happen. And just looking at these three pictures, one of my favorites is the one on the far left. Nothing, if you were to look at a science fiction movie or a TV show like Star Trek, you never see scenes like that. But those are actually duffel bags full of water. That's our water storage system on ISS. One can only imagine if a little leak happened or what's happening with the dust behind the bag. So spacecraft aren't as clean as you might think, and they're complex. There is also a lot of trash and waste control because you have people up there. And because you have people up there, you also on the far right have hygiene activity. And every time you wash your hair, water tends to travel all over the place, and you might have microbial problems. So with the potential of contamination, we monitor in flight. And we're using culture-based methodology because it's simple, it works, and it's easier to compare what we have now compared to using new methods every few years. And we check surfaces, the air, and potable water. But I know that there's going to be somebody in the crowd, and so before you ask the question, we'll ask, well, then why are you looking at some of the new sequencing technique? Because as you go deeper in space, using media-based techniques is going to be a little bit difficult and messy. And so we're looking at sequencing techniques. We have done sequencing in space. We've taken environmental sample and grown them up on media and then selected them and then compared what we got in flight to ground, and they match beautifully. And now we're switching over to what we call swab to sequencer. Basically, I don't even have to grow it on media. I'm just going to take it off a table or a surface and go ahead and directly process the DNA. So we're on the cutting edge with a lot of this. And probably when we go to the moon, we will make our shift from media to straight DNA sequencing. Oops, sorry. Now, everybody comes back after a while when they see pictures like this and they go, well, what's in the ISS? And maybe you've heard news stories talking about all sorts of horrible things. Well, I hate to break it to everybody, but when we do monitoring on ISS, what we find is the exact same thing you would find in your home. And that's a good thing. I'm rooting for the astronauts. But it's also important to note that doesn't mean it's perfectly safe. A lot of opportunistic pathogens like Staph aureus, Bacillus cereus, they can still cause infection, cause problem. Other species like Klebsiella, Enterobacter enterococcus are also identified through ISS. And so there are still organisms that could be a problem. Same way with fungal organisms. Again, we see little bits of contamination like the one in the picture, but we start to find the same things like Aspergillus and Penicillium all the time. No big deal. That's what you'd find in your home. But within that are groups like Flavus, Niger, and Fumigatus. Even we found Stachybotrys chartarum, which is a black mold we have that's very toxigenic. And we found that pre-flight. So we do like to keep up with monitoring and housekeeping to make sure that nothing gets out of control. And in case you're wondering, the water is not sterile we have in flight, but the organisms you're looking at, Ralstonia, Burkholderia, cupravitus, are the same things you'd find in a potable water line or maybe a DI water line. And so it's a really safe environment. So the next time you see an article that comes up and they say, we found this one new article, is the crew in danger? We do research on this all the time. And there's a new organism in the room you're sitting in. You just haven't had a lot of people searching for it yet. And so there really isn't a health concern from the surfaces that we've been able to find. Now, that doesn't mean things don't happen. NASA loves to call these, instead of contamination events, they always use the word anomalies. I think it makes better news coverage. But when we were on the Russian Mir space station and working with our Russian colleagues, they had basketball-sized blobs of water that would build up behind panels. And when we took a look at those, we found things we hadn't found before, like E. coli, Serratia marcescens, even Legionella species and protozoa. And that gave us an idea of, okay, we really need to control a lot of things like the water. And as much as we try to, and we do a good job, we still get contamination. And on the surfaces, it's usually due to uncontrolled water. And by that, I mean things like condensate buildup, or maybe there was a leak in a little bag and it kind of made things moist and fungus grew. We've also had air contamination, unpleasant odors. Those happen too. The hard part is those are really hard to find the source. Luckily, the HEPA filtration system I mentioned before does a great job of cleaning it up. And finally, biofilm formation in the water lines. When bacteria will grow, they'll make this gooey gel inside water lines, and sometimes they'll clog the lines. NASA's already had a pump failure because of bacterial buildup in the water line. So these things do happen. So to give you an idea of what it looks like and what my job entails, the first one on the far left is actually contamination that happened in one of the ISS modules. The crew was doing hygiene and you'd have wet towel. The material on that surface in the module is like carpet for noise abatement. Now imagine what happens when you take your wet towels and strap them to the carpet over time, and the brown spot where you see the arrow is actually mold growth, because that's what happens. So we had a lot of lessons learned there. If you look in the middle at the top, that's what a bacterial biofilm looks like, and we've had our share of problems with those as well. They took out our HEPA filters out of certain modules for a period of time, and while we never found the source, what you were looking at in the middle at the bottom is an air sample. And normally you get 1 or 2 contaminants on something like that. Plates we were getting were completely black. We were getting complaints from the crew about itchy noses and irritation. We put the HEPA filters back in, the problem went away within a week. And you may also hear about fungal or about plants being grown on ISS. And what you'll find is if there's enough water, fungus loves to grow on plants. They just do. And we are constantly trying to battle how can we grow plants without turning it into a fungus farm. But the one I want everybody to remember is in the lower right-hand corner. And in your mind, I wish I was there real time, in your mind, think, what is that? Because that's what they asked me. They showed me a picture, and I've blown it up here, and it just popped up, and the crew member said, I don't know what this is. Is it biological? Is it possibly bacterial, fungal? What do we do? And I said, I don't know. I don't have a clue. They said, do you want a sample? I said, yes, please. Unfortunately, in the time it took me to say, yes, I'd like grab that sample and bring it back, they'd already wiped it up and thrown it away. And since I don't like sending the astronauts into the garbage can to dig stuff out, we don't know exactly what that is. But I like showing it because it shows the kind of day-to-day thing that the crew runs into. And the question is, do we do a research study on it, or do I just take a rag and wipe it up? And so those are practical questions, but for the life of me, I really don't have a clue what that is. I still think it was part of breakfast or something, but I'll never know. Okay, shifting away, we've talked a lot about how rockets are built, the kind of operations and problems you run into, but as much as I'd love to tell you I know everything, I don't. We know only a fraction of what's going on up there because microbiology research has shown us that there are changes in microorganisms that occur over time. Comparing flight to ground, we've seen changes in antibiotic resistance, biofilm formation, bacterial and fungal growth rate, bacterial virulence, bacterial and fungal gene expression. And the kind of hardware you see doing this work, an example is in the upper right-hand corner. That's just one of the many bacterial and fungal processing units. We also have a unit on the ground that we use, and there are a few of them that try to mimic a lot of the environmental conditions that you may see. You can't simulate or you can't create microgravity, so you try to simulate some of the condition. And it's great for predictive and follow-up studies, and we use both, but it's still different than what you'd find with true space flight. Now, to give you an idea, and I use this particular experiment because it really transitioned us a lot when we switched from a shuttle mentality to an ISS mentality, done by an investigator named Cheryl Nickerson, currently with Arizona State University. But her goal was to take the pathogen Salmonella typhimurium, looking at it in flight and identical samples on the ground, and compare gene expression to compare disease potential in a mouse and to look at changes in cellular morphology. And we weren't expecting a whole lot. We were expecting something. We didn't expect all of what we found. For example, in the flight samples, there was a biofilm gooey material that kept showing up. that we didn't see in the ground control. And importantly to NASA, a lot of, or the in-flight grown Salmonella typhimurium killed mice faster and killed them at lower doses than identical bacterial culture grown on the ground. About a 2.7-fold difference. So this really got a lot of attention. What got less attention, but was more important to me, was looking at a lot of the gene data. And what we found is genes were being changed all over the genome. The proteins being produced were changing massively, but 167 genes were differentially regulated by spaceflight in a number of areas, and one of the regulators, a master molecular regulator of RNA, HFQ, was identified basically controlling about 33% of the genes we found differentially regulated. Now, this was important because up until this point, we didn't know if you took bacteria or fungi in space, you'd kind of figured they didn't have a microgravity receptor. So would they just be confused? But from looking at this, it's sort of like, no, they think they're doing something in a mechanistic way. It's really an important study. So important that NASA, still worried about the virulence, said, you got to fly that again. I mean, and like now. And so they said, redo that disease potential aspect. And by the way, to give you an idea, the second study validated the first study. But we were also lucky because we got to, you had a few chambers that were still empty. So we did some experiments with media composition as well. And what we found, and please bear with me because I know this can get really confusing because there's a lot of data here. Basically, if you look at one shuttle mission on STS-115 and the other shuttle mission on STS-123, you saw the same pattern when we were using the medium called LB. It's Linux broth, for those who are curious. We also took a look at a minimal salts media, basically low organics, low proteins, primarily just a little bit of glucose and a lot of salt. When we did that, we didn't see the change in virulence. We also added those salts to the LB media, making a specialized media you'll see called LBM9 salts. That one didn't increase in virulence either. So we knew that something was going on with the salts that was regulating this effect. It was interesting because at this point, for those people with a micro background and an infectious disease background, you're going to go, yeah, we know the media type changes the virulence profile. We've known that for years. But if you will look between what happened on the ground and the changes between the media type with 1.4, 5.7-fold differences, that same experiment done in space showed a 10-fold increase, showing a 12.8 and 56.8. Bottom line, whatever makes a difference with media type was being exacerbated tenfold in spaceflight. And we're still trying to figure out all the details of why that happened. And if you'll take a look at all of these, because like I said, we're not the first to show changes in microorganisms, but the question is why? And it's hard because it's really not conclusive, but what we've been kind of agreeing on is what you're seeing in spaceflight isn't a direct response. There's not little gravity sensors in the microorganisms, but it's an indirect response to maybe hydrodynamic pressure or convective forces. And comparison between all of the spaceflight data, because this is the one I usually get asked, well, if you've done 100 experiments, why don't you just take all that data and merge it in together? Well, we use different organisms, different media types, different aeration levels, different flight hardware, and different microbial species. So making that comparison is really, really difficult. So now we're going to get into a postulation, and hopefully you guys like this because this is the first time I'm trying this. What do you think it is? Well, there are certain groups right now. The two leading candidates it's mechanotransductive. We're getting different shear forces on cell surfaces. They're responding differently, and we've got evidence that shows that. But there's also a good argument about extracellular mass diffusion. Maybe I had to add all of those ions to change what was going on because they're not diffusing well in a quiescent microgravity environment. It's also very credible. It's also possible it's a combination of these two, or maybe not really either one exactly. One other piece, and I'd mentioned HFQ, in looking at the mechanism, we saw it in Salmonella, but then we again saw that same kind of high proportion being regulated by HFQ in Pseudomonas aeruginosa, Staphylococcus aureus, and one investigator found it in Vibrio fischeri. So that probably is a player in what's going on. We still don't have all the details. So without going into what is truly a huge number of current research projects, I wanted to just bring out a few just to let you know kind of what we do and what's going on. One of the experiments we're doing right now for our human research program is they said, okay, can you see how widespread these changes are by looking at the ground analog and saying, okay, I'm going to run 5 different pathogens in it. And we're taking a very drilled down close look at these, trying to see if there's any similarities between Salmonella typhimurium, Pseudomonas aeruginosa, Salmonella enteritidis, Burkholderia cepacia, and strep pneumo. So we're still working on that one, getting a lot of good information. How well it all integrates and gives us a mechanism, we'll wait and see. One of my colleagues, Bob McLean, is working on a biofilm project because, normally we run one organism in these things. Bob's an expert in the field and is actually looking at a couple of the organisms that might be up there and is looking to see how they change their architecture or how they're built, how well disinfectants work on them, and how much corrosion they cause, or is it different in flight than on ground? Cheryl Nickerson is also finished up an experiment on host pathogen interaction and actually did infection of human cells in flight. And it was a beautiful article that came out on that one from Jen Barilla et al. In 2021. A couple more, and one of them is another one from Dr. Nickerson where she's looking to see if infection changes with the addition of a lunar dust simulant. We're going to the moon. Does that actually change the potential for infection if you're going to be breathing and getting around lunar dust all the time? And one more that I always like to mention because it's a popular question is, do we ever do microbiome studies? And the answer is we started doing microbiome studies right around 2010. And one of the first big ones was by Dr. Hernan Lorenzi out of JCDI. And he completed one on 9 astronauts. Now, that doesn't sound like a lot for people who have done microbiome studies. It's a lot of astronauts and a lot of time points and a lot of sites, including skin, nostril, fecal. They were collected pre-flight, in-flight, post-flight. Great article came out of it by Alex Voorhees in 2019. But this started a lot of microbiome studies. People are starting at it into their research. NASA's trying to get it done on almost every crew member. We believe there's a lot of good, valuable information in there. It's just going to take a while to get to a substantial number to make those kind of analyses. And if you look on your right, and if you ever wonder how in flight they take a fecal sample, after the defecation, and you can see with a little yellow tennis ball in there, we take a long swab and get a sample and pull it out. It's not the most popular thing to do in flight, but it's giving us a great amount of information. And so as I'm bringing a lot of this to a conclusion, I wanted to toss out, I've gone through so much, so fast. I apologize that I'm having to give an overview because there's so much information left. But for those who are interested in the topic, we've got a review that is probably as comprehensive of a review as I've ever written on what's going on that will be coming out in microbiology and molecular biology reviews. They told me the target date for release is sometime in September. So that might help out if you're interested in the topic. And so with that, are there any questions?
Thank you so much for that fantastic presentation. That was really awesome. And I'm sure that we all learned heaps from that. Okay, questions. Surely there are some questions.
Yeah, I've got a question for Dr. Art. So it seems that the space environment seems to accelerate changes in microorganisms, and I guess it's in somewhat unpredictable ways. Is there any sort of what do you feel is like the most likely explanation, I guess is the question.
And that's a great question. And then I went in with all the mechanisms. Right now, my hypothesis, my belief, it's a combination of effect. But what you're doing is, one can imagine the bacteria have never been in outer space before. Almost guarantee that. And so why would they have any mechanisms that would adapt to outer space? And the answer is probably not adapting to outer space. They're probably thinking they're in an environment that is reflected by what they see in space. One of the ones we thought about is something like the brush border, the intestinal villi, where you have a very quiescent, low shear environment. It makes sense if the bacteria is coming in through the intestine, get into the brush border, and go, wow, the shear levels are really low. I think it's time for me to start looking at becoming a pathogen and invading. And so what you're doing is you're tricking the bacteria into becoming hotter or more virulent or turning on mechanisms that it normally wouldn't. The confusing part and the confounder is it's only seeing certain things are being seen, other things are not being seen, and that may be why it's kind of a mixed bag. Because a lot of the genes normally associated with virulence weren't increased, weren't upregulated. And so it's fascinating for two reasons. Number one, we found a new mechanism for increasing virulence without triggering a lot of the normal gene expression. And number two, now you're looking at what triggers each one of those genes. So it sets up a very interesting kind of situation. But to answer your question, I'm sorry, I can talk on this all day. But to answer your question, I really think the bacteria think they are in certain environmental conditions that would promote what they would normally do in those conditions.
Yes, yeah, thank you.
I had a question as well, just sort of building on what Loshi just asked, but given that the bacteria and everything is evolving so much, do you think into the future, the biggest threat is going to be things that do currently exist on Earth, given that a lot of that's being found in that environment? Or do we think that it would be sort of novel threats emerging as spaceflight becomes a bigger deal?
It's a good question. I believe that a lot of what we're going to have to worry about in the near future are things we bring with us. I think as you start to look longer term, then the question comes up, and we haven't done a lot of research into it. Okay, there's a selective pressure that may change these microorganisms over time. So first it's don't take salmonella up there and put it in your food. Then it's, well, if salmonella got up there, what's it going to look like after it's been up in space for 20 years? By the way, I'm not a big believer that you're going to convert normal plain Jane bacteria with no virulence mechanisms into evil creatures that you see in science fiction movies. But then you also get into, and I'll throw this in just to be controversial, what are we going to find if it's not adapted during space flight and we start colonizing Mars? What are we going to find? And even if we don't find something, how are things going to adapt to the new environment? And those are, and I don't have any idea. We haven't even started that kind of research because we really haven't been pushed that hard to do it. But as far as long duration changes due to microgravity, you're going to find some. Whether they create a more virulent organism or what, we don't know. Did that kind of answer it?
Yeah, thank you.
Any other questions?
I had a quick one that might be sort of tangentially related, which is, have you looked into the gut microbiome of astronauts after they've come back from spaceflight? And have you noticed any dysbiosis or any changes like that?
The answer is we have looked into it. The hardest part on that is getting a representative population. The article that I mentioned before that came out in 2019 by Voorhees et al., we saw trends. But when you're talking about trends of 9 people, it's not overwhelming. Because then you get into things like, is it a spaceflight effect? Or is it the fact they just started eating spaceflight food on Tuesday and now you're seeing a shift? There was an experiment done and it had so many advantages and so many different advantages. Does anybody remember when they sent up Scott Kelly in flight and his twin brother, Mark Kelly, was still on the ground? They did a lot of testing, including microbiome testing. It was exquisitely hard to distinguish because they weren't eating the same diet. They were doing a number of things. To get a large enough population, and NASA's working on this now, saying, okay, you're eating spaceflight food, what are we seeing? And by the way, that includes the person who likes the shrimp cocktail and eats it every night versus the person who likes the beef stroganoff and eats it every night. So it's a challenge. I think we will get a better picture in time. But to answer your question, yes, we've looked at it. Yes, we have seen trends with an incredibly small n. And no, we probably don't have a lot of really good answers yet. Probably in about 10 more years with the rate it's going, we'll start to see more where we can say, aha, that's a spaceflight effect.
Thank you.
I might ask a question while we're waiting for other people to think, and that is, how much of a risk do you think that the combination between the potential increased virulence of some pathogens and the immune dysregulation that humans experience during spaceflight, is that sort of a perfect storm for or susceptibility to infectious disease, or is it something we really don't need to worry too much about?
The answer is, because you can go back to some of my old presentations and I use that exact same phrase, are we looking at a perfect storm? And the reality is it could be, because if you'll look at a lot of the immune research, they have been able to adjust and get rid of a lot of the dysregulation we see by having the crew in a normal sleep cycle, exercise regularly, have good nutritious meals, trying to take the stress away, so to speak. But if you can't do that, you'll have a highly stressed population with a dysfunctional immune system. The microorganisms, if I can prevent salmonella from getting up there, I probably don't have a risk with salmonella, even though it would change possibly its virulence level. And so could you put it together for a perfect storm? And the answer is yes, you really could. I'm personally doing a lot of research right now, looking at a lot of the statistics associated with infectious disease to try to say what's going to be up there, what are we afraid of changing, and bottom line, I don't want salmonella up there anyway. And the fact that it's twice or sevenfold more virulent and the immune system's more susceptible, probably I'm going to be very conservative on my monitoring, but it's still going to be a very bad day in space either way. Does that make sense? And by the way, just as a quick aside, so I can gross everybody out, after this is over, as you're going home or as you're walking through classes, think to yourself, what would a good bout of food poisoning look like in microgravity? And that's one of the reasons why I work really hard to make sure that would never happen.
Yeah, that's a really good point, and would be fairly horrendous to have to think about. And it's actually one of the things that people often mention when they're talking about infectious disease in space is if somebody does happen to come on board with an infection or somebody develops a gastro in space, then since you're in such a confined environment, how do you actually isolate somebody and prevent the rest of the crew getting infected? So do you have any wonderful answers to that conundrum?
We talked a lot about it, especially during the COVID epidemic, because it was one of those, people on the ISS were safe, but we were still sending people up there. And you almost come to the conclusion of eventually you're going to expose everybody. And that's an unfortunate situation, which makes the prevention that much more important. Other things like food poisoning are fascinating. And again, forgive me if I'm getting just a little too descriptive here. Crew member A ate several items, including the shrimp cocktail, beef stroganoff, and some specialized beef jerky from France. Now, if that person gets sick with food poisoning, can someone else come in and eat any of those items? And that's one where, well, it's not sequential, but you don't want to take a risk and say, well, for the last 3 meals or for the last 2 meals, this is what that person had. So now you're taking part of the food supply away because you really don't want them eating something that could already be contaminated. So there are certain actions you can take, but I won't lie. It's really challenging. If you're having diarrhea and vomiting uncontrollably that will be contaminated with a pathogen, how do you prevent exposure to other crew members? And those are just practicalities that I think about too much, but those are things you really have to think about.
Yep, thank you. Any last questions?
I had one question for Dr. Wright. Thank you for giving the talk. This question is, I don't really expect it to have a very straightforward answer, but just to get a gauge at where you think humanity's understanding of, microbiology is at for our preparation to get to Mars. I wanted to pose a quick hypothetical. So let's say tomorrow you woke up and everything else in terms of planning the trip to Mars was ready. Like the spaceship was ready to go, everything else was sorted out. Would you personally feel comfortable with humanity's current knowledge of, space flight and microbiology to send astronauts to Mars? Or do you still think we need a few more years?
There are certain aspects, and believe it or not, this is, I see where you're coming from, but to me, it's easy to answer. There are certain aspects that I believe are really well researched, are really well done. Things like what kind of organisms do we find in habitats? Those I have no problem, and I think we are good to go for Mars. There are other areas, though, with infectious disease, changes in microorganisms over time and how they evolve. If they evolve, does that change infection? And I'm going to be giving another presentation this coming Thursday to basically tell my management, no, we don't know enough. I really don't think we know enough. We can go to the moon, but only because we can get back pretty fast. Going to Mars, I think there are a number of different things where we really need to do more research before I would feel very comfortable. It's easy for me to answer because if I felt comfortable, they'd go, great, you don't have to do any more research. And the answer is no. There are certain questions we have that are just, and well, we do a green, yellow, red, and I've got questions on Mars that are red. So I would say now those aspects, no.
Okay, thank you so much.
And after watching the National Geographic TV series Mars, and I know it was all for dramatical effect, but they found a novel pathogen in the water on Mars, and then that wasn't particularly well contained when they were looking at it in the lab, and then it got out and killed half the people who were there. So I imagine that whole issue of biocontainment of any samples that they might bring on board too would be a fairly serious thing to have to consider.
It is, and it's kind of fun because I almost, and on so many series, we'll see, well, we're going to deal with infectious disease this episode, but it's real. A lot of those things are real. Like you're saying, are they a little overly dramatic? Probably so. But yeah, an outbreak somewhere on Mars would be a mess. And that just, and when we're dealing with, in our world, we deal with what we call loss of mission, loss of crew. You'd hate to see either one of them happen. Even if they're going, well, we can't get any work done for a couple of weeks. That's a huge cost impact on what's going on. So yeah, it's one we're trying to avoid.
Yeah, exactly. Thank you very much. Well, I think we're out of time, but thank you very much indeed for coming along today. It's been an absolutely fantastic session. I've really enjoyed it, and judging by the questions everyone else has as well. And we really appreciate you taking the time out of your Sunday evening and sharing all your knowledge and expertise with us. So thank you very much indeed.
Definitely my pleasure. Thank you for the invitation.
Hopefully we'll see you another time.
That's a deal. And if any other questions pop up, feel free to send them to me. I'm happy to answer.
Oh, that's awesome. Thank you so much.
And thank you.